Hello everyone, my name is Laura Gargan and today I'm very happy to present a collaboration between UCD, the University of Crete and the Hellenic Centre for Marine Research. This work is entitled Bacteria Are Everywhere, Even in Your CO1 Data, aka The Art of Getting to Know the Unknowns and Shine Light on the Dark Matter. I'll just begin by giving a quick overview of what I will speak about today. Firstly, I'll briefly speak about CO1 within the context of eDNA metabarcoding. I'll describe our methodologies relating to the development of a pipeline in order to determine the unknowns in CO1 data, and I'll also discuss some of the results that we have found using some example data sets. Finally, I'll discuss some conclusions of our project. In metabarcoding studies, primers often target the mitochondrial CO1 gene. You can see in the image on the top right-hand side of the screen that there are many primers that have been designed to amplify this gene, and these are just a few examples. And this is for a good reason, because CO1 has many of the desirable properties of a universal marker, and it's been widely used as a barcode for species identification. And this has earned it the title of the barcode of life. There are huge and growing repositories for CO1, and a great example is the BOLD database, which currently has more than 9 million barcodes. Of course, there's also GenBank and curated versions of the CO1 that are available in GenBank, such as the Midori database, and these are also commonly used. However, even with the availability of these great repositories and the increasing efforts of researchers that are involved in barcoding initiatives across many taxonomic groups, a great number of sequences from such studies do not match reference sequences and are often referred to as dark matter. And for this reason, the aim of this study was to discover the origins and identities of these CO1 dark matter sequences. So with that end goal in mind, we developed a pipeline that would allow us to easily place our unknown sequences onto a phylogenetic tree containing 3 important domains of life: the eukaryotes, the bacteria, and archaea. We built a reference phylogenetic tree that included as much CO1 sequence information across those 3 domains. An overview of the steps that we followed is shown in the figure on the right. We retrieved over 180,000 eukaryote sequences from the Midori 2 database. And then we used the API of the BOLD database to retrieve bacteria and archaea sequences. Consensus sequences at the family level were constructed from each of these initial CO1 datasets. These were then combined, and the reference phylogenetic tree for CO1 was built using over 1,200 consensus sequences, with more than 80% of those coming from eukaryotic taxa. We were then able to place our query sequences onto the phylogenetic tree to determine the origins of our query sequences. In relation to the query sequences, we had 2 datasets that were available from pre-existing metabarcoding projects. These datasets were generated through Illumina MySeq efforts. One of the datasets originates from marine eDNA samples and the other from freshwater samples. You'll be able to see in the table the methods that were used to amplify and perform taxonomic assignment on each of the datasets. You can see that for the marine dataset we used degenerate CO1 primers, and in the freshwater dataset we used Fulmer primers. From the clean sequences, we have almost an order of magnitude difference between the number of ASVs found in marine and freshwater datasets, with a much higher number of ASVs occurring in the marine dataset. For both datasets, the ASVs were blasted against the Midori unique database, requiring at least 97% identity between the reference and the query sequence. In the end, only 1% of marine samples were assigned taxonomy, and 10% of freshwater were assigned taxonomy. So in terms of results, I will firstly present the results from the phylogenetic placement of our marine water samples. I should note that these placements are all represented on chronoplots for ease of visualization. So we'll see each differently coloured portion of the plot represents a different domain, and the domain containing the highest proportion of query ASVs is coloured in red. In this first plot, you'll see this is where we placed all of the query sequences from our marine ASVs. Here we find that approximately 45% of all of our query ASVs are of bacterial origin. The remaining ASVs are pretty much equally shared between the archaea and eukaryotes, as well as what is described here as distant, which you'll see coloured in the purple part of the plot. These distant assignments are those that didn't fit into any of the included domains, and so we believe it's possible that these could represent things like viruses or maybe sequencing errors. Also recall that only 1% of these ASVs were assigned taxonomy by blasting the sequences against the Midori database. If we look at the second figure where we finally phylogenetically placed the assigned ASVs onto our reference tree, you can see that the majority of ASVs are eukaryotic in origin, but we still have some bacteria and archaea, as well as quite a few assignments placed on this distant clade. The last plot that I'll show for the marine sample shows the dark matter or the unassigned ASVs. And as you can see, the plot doesn't look so different from the first plot, and this is because only 1% of the ASVs were assigned. So basically, almost all of our data set is dark matter and can be represented by this plot. The next set of results applies to our freshwater data set, where a better, but still not great, proportion of ASVs were assigned taxonomy by blasting against the Midori database. Firstly, if we again look at all of the ASVs present in the freshwater dataset, we can see that the majority of ASVs are of eukaryotic origin, with only a tiny number of archaea, which isn't really visible in this particular plot because it's so small, as well as a small fraction of ASVs belonging to this distant clade, represented by blue in this plot, and about 30% belonging to bacteria. In the second plot, we can take a closer look at the 10% of the ASVs that were assigned taxonomy in the freshwater dataset. And we can see that we still have some ASVs whose origins are not eukaryotic. And this brings up an interesting point regarding our original assignments for both the marine and the freshwater datasets, really. And that's, why do we see these samples, these placements for ASVs that were assigned taxonomy to eukaryotic species? One reason might be that our assignment parameters were not sufficiently stringent, and so we're seeing evidence of possible misassignment. However, I would also point out that we did not intend this tool really to be a replacement for taxonomic assignment. It's simply a way to ascertain the very broad origins of ASVs in CO1 metabarcoding. In this third figure for the freshwater samples, if we look at the proportion of ASVs belonging to each domain for our unassigned sequences or our dark matter, we see a similar pattern for all of our ASVs in the freshwater dataset, where the majority of ASVs are eukaryotic in origin, followed by bacterial placements. Regardless of whether we look at the marine or the freshwater samples, and their variable proportions of bacterial placements, it's still clear that ASVs of bacterial origins contribute a large amount to the CO1 metabarcoding data. On that note, I think we can conclude that bacteria are everywhere and they are in your CO1 data. We tested our approach using different data sets, generated using different CO1 primers that have been commonly used for animal DNA metabarcoding studies. We believe that while other datasets generated using different sampling protocols, primers, and bioinformatic pipelines may produce different patterns to what we have presented here, it's likely that all datasets will contain bacteria to some extent due to their ubiquitous nature. In relation to the difference in proportion of bacterial ASVs in our freshwater versus our marine eDNA samples, a good question might be, is this the result of sample type or our primers? And my answer to this would be that we might expect greater diversity in general in marine samples, not at least because marine samples contain DNA originating from terrestrial, freshwater, and marine sources, but we would also strongly consider the influence of primer choice on the number of bacterial ASVs. For the marine samples, we used degenerate bases in the primers, and this might have contributed to the amplification of many more non-eukaryotes in those samples. As I mentioned previously, we don't intend this tool to be used as a replacement for a taxonomic assignment, but really as a way for researchers to shed light on the origin of the unknown dark matter that might be lurking in their metabarcoding data. For this reason, we also recommend that bacterial CO1 sequences are included in reference databases that are routinely used for taxonomic assignment, so that we can be aware of the proportion of bacterial and other non-target organisms that might be present in metabarcoding datasets. In this way, we might be able to direct efforts towards protocols that reduce their prevalence. Finally, we aim to make our pipeline and our reference tool publicly available for other researchers who might wish to shine a light on their unknown unknowns, and this will soon be made available as a Docker image. Before we finish, I would just like to acknowledge on behalf of my collaborators and I the computational resources that were provided through the IMBBC, as well as funding through the HFRI and the GSRI, as well as special thanks to Kazutaka Kato of the MAF team for helping us to overcome some massive obstacles with the alignment of our many reference sequences. And of course, a very big thank you to the organisers for allowing us the opportunity to present our work here today, and a thank you to everybody for attending and listening to our talk.